Could the intricate tapestry of human memory and skill ever be directly transferred, bypassing years of learning and experience? While the concept of uploading knowledge remains firmly in the realm of science fiction, a rigorous scientific exploration of its underlying principles reveals fascinating insights into the brain's profound complexity and the cutting edge of neurobiology. This discussion delves into the current understanding of memory encoding, the nuanced role of epigenetics, and the ethical quandaries posed by potential genetic cognitive enhancements, all through the lens of contemporary scientific inquiry. The notion of instantly acquiring complex skills or vast datasets of memories is undeniably captivating. From a scientific standpoint, this concept challenges our fundamental understanding of learning, neural plasticity, and the very essence of individual identity. It compels us to dissect the biological mechanisms that underpin cognition, pushing the boundaries of what is currently known and what might theoretically be possible. Current scientific consensus firmly places direct, complex memory or skill transfer in humans outside the realm of present-day capabilities. Our understanding of how memories are encoded, stored, and retrieved is still evolving, revealing a system far more intricate than a simple data repository. This complexity necessitates a deep dive into the molecular, cellular, and systems-level processes that govern memory formation. Memory is not a monolithic entity, but a dynamic, distributed process involving vast neural networks across the brain. Encoding, the initial acquisition of information, transforms sensory input into a format the brain can store. This process is highly active and constructive, influenced by prior knowledge, attention, and emotional state, shaping how new information integrates into existing cognitive frameworks. Storage refers to the maintenance of this encoded information over time, ranging from fleeting working memory to enduring long-term memories. Retrieval, the final stage, involves accessing and reactivating stored information when needed. These stages are not discrete, but interact continuously, with retrieval often leading to reconsolidation and potential modification of memories. The hippocampus, a seahorse-shaped structure in the medial temporal lobe, plays a critical role in the formation of new declarative memories, facts and events. It acts as a temporary hub, coordinating the initial encoding and consolidation of memories before they are gradually transferred to more stable cortical regions for long-term storage. Damage to the hippocampus often results in anterograde amnesia, the inability to form new memories. Beyond the hippocampus, various brain regions contribute to different memory types. The prefrontal cortex is crucial for working memory and executive functions, while the amygdala is central to emotional memories. Procedural memories, such as riding a bicycle, are largely mediated by the basal ganglia and cerebellum, highlighting the distributed nature of memory systems. At the cellular level, memory formation is underpinned by synaptic plasticity, the ability of synapses, the junctions between neurons, to strengthen or weaken over time. Long-term potentiation, LTP, a persistent strengthening of synaptic connections, is widely considered a primary cellular mechanism for learning and memory. Conversely, long-term depression, LTD, involves a weakening of synaptic efficacy, contributing to memory refinement and forgetting. These synaptic changes involve complex molecular cascades, including the activation of specific receptors, for example, NMDA receptors, protein synthesis, and gene expression within neurons. The precise patterns of neural activity and synaptic modifications form what are hypothesized to be engrams, physical traces of memories in the brain. Recent advancements in neuroscience, such as optogenetics and chemogenetics, allow researchers to identify, activate, and even suppress specific engram cells in animal models, offering unprecedented insights into memory circuits. Epigenetics refers to heritable changes in gene expression that occur without altering the underlying DNA sequence. These modifications act as crucial regulatory layers, influencing which genes are turned on or off in specific cells and at particular times. In the context of memory, epigenetic mechanisms play a profound role in neuronal plasticity and the consolidation of long-term memories. Key epigenetic mechanisms include DNA methylation, histone modifications, and the action of non-coding RNAs. DNA methylation, typically occurring at CPG sites, involves the addition of a methyl group to cytosine bases, often leading to gene silencing. In neurons, dynamic changes in DNA methylation are observed during learning, influencing the expression of genes critical for synaptic function and memory consolidation. Histone modifications, such as acetylation, methylation, and phosphorylation, alter the accessibility of DNA to transcription machinery. For instance, histone acetylation generally loosens chromatin structure, promoting gene expression, while decetylation compacts it, repressing transcription. These modifications are highly dynamic in neurons, responding to neuronal activity and contributing to the rapid gene expression changes required for synaptic plasticity and memory formation. The interplay of these epigenetic marks allows neurons to fine-tune their gene expression profiles in response to environmental stimuli and learning experiences. This dynamic regulation is essential for the long-term changes in synaptic strength and neural circuit remodeling that underlie memory storage. For example, specific histone modifications have been linked to the consolidation of fear memories and spatial memories in animal models. While epigenetic mechanisms are undeniably crucial for an individual's memory formation, the concept of inherited memories through epigenetics is often misunderstood. Transgenerational epigenetic inheritance, where epigenetic marks are passed down across generations, has been observed in some model organisms for certain traits, particularly in response to environmental stressors. However, these typically involve broad predispositions or metabolic changes, not the direct transfer of complex specific memories or learned skills. The scientific consensus is clear. While epigenetic changes can influence an individual's susceptibility to certain conditions or modulate behavioral tendencies, they do not equate to the direct inheritance of specific, detailed memories or learned information. The intricate neural networks and synaptic connections that define a memory are formed through an individual's unique experiences and learning processes, not pre-programmed through germline epigenetics. Distinguishing between the inheritance of a predisposition and the inheritance of a memory is critical for accurate scientific discourse. The advent of advanced gene editing technologies, particularly CRISPR-Cas9, has opened unprecedented avenues for manipulating the genome with high precision. This raises the theoretical possibility of genetic cognitive enhancement, where specific genes associated with cognitive functions like memory, learning, or intelligence could be targeted for modification. Such interventions could potentially be pursued through somatic gene therapy in adults, or, more controversially, through germline editing or embryo selection. Research has identified numerous genes implicated in cognitive processes. For example, genes involved in neurogenesis, the birth of new neurons, synaptic plasticity, for example, genes encoding NMDA receptors or AMPA receptors, neurotransmitter synthesis and reception, for example, dopamine, serotonin pathways, and neurotrophic factors, for example, BDNF, brain-derived neurotrophic factor, are all under investigation for their roles in cognition. Modulating the expression or function of these genes could theoretically enhance specific cognitive domains. However, the genetic basis of complex cognitive traits like intelligence and memory is highly polygenic, meaning they are influenced by hundreds, if not thousands, of genes, each contributing a small effect. This polygenicity makes targeted genetic enhancement incredibly challenging and unpredictable. Altering one gene might have unforeseen pleiotropic effects on other biological systems or cognitive functions, given the intricate interconnectedness of genetic networks. Embryo selection, where embryos are screened for specific genetic markers and only those deemed desirable are implanted, is another potential pathway for genetic enhancement. While currently used for preventing severe monogenic diseases, its application for complex polygenic traits like intelligence raises profound ethical and societal concerns. The predictive power for such traits is currently very low, and the practice could exacerbate social inequalities and lead to a designer baby paradigm. The scientific community approaches genetic cognitive enhancement with extreme caution. The complexity of the brain, the polygenic nature of cognition, and the potential for unintended consequences necessitate rigorous research and a deep understanding of gene-environment interactions before any such interventions could be considered safe or effective. The ethical implications, as discussed next, are equally formidable. The prospect of manipulating memory or enhancing cognition through genetic or biological means raises a myriad of profound ethical considerations that demand careful deliberation from the scientific community, policymakers, and society at large. These issues extend far beyond mere technical feasibility, touching upon fundamental questions of identity, autonomy, justice, and human flourishing. One of the foremost concerns revolves around identity and selfhood. If memories can be altered, implanted, or erased, what happens to an individual's sense of self, their personal history, and their unique narrative? Memories are integral to who we are. Manipulating them could fundamentally reshape personality, values, and relationships, potentially leading to psychological distress or a loss of authentic self. The concept of informed consent becomes incredibly complex when dealing with interventions that could alter the very capacity for consent or self-perception. Safety and unintended consequences are paramount. The brain is an extraordinarily complex organ, and our understanding of its intricate networks and molecular pathways is still incomplete. Any intervention aimed at memory or cognition carries a significant risk of unforeseen side effects, including neurological disorders, psychological disturbances, or alterations to other cognitive functions. The potential for irreversible harm necessitates an extremely high bar for safety and efficacy before any such technologies could be considered for human application. Equity and access present a significant social justice challenge. If advanced memory or cognitive enhancement technologies become available, they are likely to be expensive and accessible only to a privileged few. This could exacerbate existing social inequalities, creating a new form of cognitive divide between enhanced and unenhanced individuals. Such a scenario could lead to a two-tiered society where access to opportunities and resources is further stratified based on biological enhancements, undermining principles of fairness and equal opportunity. The potential for misuse and control is a chilling prospect. Technologies capable of manipulating memory or implanting skills could be exploited for coercive purposes by authoritarian regimes, corporations, or even individuals. Imagine the dystopian implications of forced memory alteration, the implantation of specific ideologies, or the creation of perfect soldiers or workers through biological programming. Robust ethical guidelines, international regulations, and public oversight would be essential to prevent such abuses. Finally, the very definition of human flourishing comes into question. Is there an inherent value in the struggle of learning, the process of overcoming challenges, and the organic development of skills and memories? If these processes can be bypassed, what does it mean for human effort, resilience, and the intrinsic satisfaction derived from earned achievement? These philosophical questions underscore the need for a broad societal dialogue, not just a scientific one, about the ultimate goals and implications of such powerful technologies. While the direct uploading of memories remains a distant, perhaps impossible dream, the scientific journey to understand memory, epigenetics, and genetic influences on cognition continues at a rapid pace. Current research frontiers are focused on deciphering the neural codes of memory, understanding the precise molecular mechanisms of synaptic plasticity, and exploring the therapeutic potential of modulating these processes for neurological and psychiatric disorders. Areas like optogenetics and chemogenetics are revolutionizing our ability to precisely control neuronal activity and dissect memory circuits in animal models. Brain-computer interfaces, BCIs, are making strides in restoring lost function and even enhancing certain cognitive abilities, albeit through external devices rather than direct biological memory transfer. These advancements, while distinct from direct memory transfer, contribute to our foundational knowledge of the brain. The ethical implications of these emerging technologies must be continuously evaluated and debated. This requires a multidisciplinary approach involving neuroscientists, ethicists, legal scholars, sociologists, and the public. Proactive engagement and the development of robust ethical frameworks are crucial to guide research responsibly and ensure that any future applications serve humanity's best interests rather than leading to unforeseen societal harms. The scientific pursuit of understanding memory is one of humanity's most profound endeavors. It promises not only to unlock the secrets of the brain, but also to challenge our perceptions of self, learning, and what it means to be human.